forget the drawing. But where one forms a head, the other cells form a foot, but they have the same DNA. It's all about controlling which of these genes that are on the cells in the cells, which are turned on and which are turned off. So the foot genes are going to have a certain sequence. The head genes are going to have a different sequence. And so the foot genes might have this one, and that one turned on. And maybe there'll be more of the head genes on one chromosome versus another. Maybe there'll be more head genes on certain chromosomes, but the point is that different genes are going to be turned on in different tissues. That's called differentiation. When you first were conceived, when your father's sperm met your mother's egg and they formed you were only one cell. You were a single cell. You started to divide 1 into 2, 2 into 4, etc., until you were a group of cells. During development, you turned into a group of cells, a little ball of cells called a blastocyst. And eventually, some of these cells started to change. They started, at first, when you were just this, and probably when you were just in this range of cells, we'd call these cells omnipotent. They could become anything. This cell could become any part of you, your brain, your eye, your foot, your liver, whatever. But soon these cells started to differentiate. Soon the head cells started to come together and the skin cells started to come together, and the cells were going to turn into blood and tissue, and blood vessels and such were going to come together. And so they started to differentiate. They started to become different. How would the cells become different? They would turn different genes on and different genes off. Does that make sense? So these cells lost their ability to become omnipotent. They no longer were omnipotent. They're not necessarily completely differentiated. So at some point, they became different tissues. Part became, brain. Part became an eye. Different parts of the eye, right? Because to this organ called an eye, there's different tissues. Some became muscles. Some became nerves. So once they became these different types, these different specific types, they're completely differentiated. So it's very hard for them to be anything else. That's why once you have a muscle in your hand, somebody cuts off your arm, once you're differentiated, once you're an adult, once you're a child, you've already made these decisions. These cells have made these decisions. They're turned off and they're turned on. Genes are turned off and genes are turned on. So that cell could only be a muscle cell. It can't become a bone cell. Your arm is not an arm cell, right? Your arm is made of skin cells and blood vessel cells and blood, right? And muscle and hair follicles and bone and and cartilage, right? So there's all kinds of different cells here. It's not just one cell type. So if I cut my arm off, these cells in my body would have to turn into bone and muscle and blood and skin, and they can't do that because they've already differentiated. They've already become different, and so they've lost their ability. They're no longer omnipotent. They're completely differentiated. There are some cells that keep their a level of omnipotence. We call them stem cells. Stem cells used to be just from embryos, embryos that were miscarried or aborted or whatever. And we're usually at this stage when they're one or just a group of cells here. But these stem cells are very important. You can get some level of potential. That's what potent means, right? Potential. Omni means all-encompassing, right? Potential, so they can become anything. Well, there's a type of stem cell you can get from the umbilical cord. That's one. There are other stem cells in your body, in your bone marrow and etc., but they've already lost some of their potential. But stem cells have been a very strong source of our research because they can allow us to become any part of your body. Now, we've come up with techniques that allow us to reset the DNA. We have methods now. We've done it with sheep and with other vertebrates, which when I was your age, scientists thought were going to be impossible. They thought anything more complicated than a frog wasn't going to be able to do this. But we found that if we do a series of techniques, we can undifferentiate. This undifferentiating process does come with issues. We haven't been able to perfect it yet. We can't clone you. But the hope is that we can take one of your skin cells or a cell from your leg, and if you need a heart, we can undifferentiate it at some point in the future through whatever process, and we can then grow you a new heart. We don't want to grow a clone. What would be the problem with just growing a clone? Where's some of the problems of growing a clone of you to harvest a heart or liver or whatever you need?
Wouldn't it be like deformities and stuff after?
Could be deformities and stuff because of the techniques, but let's assume we don't get rid of any of the problems in the techniques. We can grow a new clone. What's the problem? In fact, some people think that in China, some scientists in China said they've done it. So we can clone you. We can take and make a new copy of you. And then if you get sick as you get older, we can just change out your heart or your liver or whatever. What's the issue?
Would that person, the cloned heart, grow with you?
That's a good point. You have to wait till it grows up, right? So it's going to have to wait at least 18 years, right, for it to be fully mature. So that's no good because you're not going to have that much time probably. But let's say we can speed that up. So now we grew a whole clone. What's the issue? That thing has a brain. How are you going to take its heart for you? It's a living creature. It's a different creature. It's your twin, right? A clone is your twin. You're going to kill your twin so you can live? Oh, you're harsh. You would do well in Game of Thrones. But no, that is an ethical problem, right? So, but the solution is don't grow the whole clone. Just grow the heart. You need to learn these signals, though. It all comes back to these genes. We need to learn what those genes are that need to be turned on and which need to be turned off so that we can turn the right ones on and off so we can grow you an organ without having to grow a whole twin. Does that make sense? All right, so that would be the ultimate goal. By the way, this explains why a lizard can regrow a tail or a leg or an arm. Because lizards and geckos, for instance, and I don't know how many toes they have, but let's just say they look something like this. I know they don't, but whatever. So a gecko, you cut the gecko's arm off, it turns out that the gecko has some stem cells at the base of their arm and their tails. And if you cut them up, they have stem cells. They have that potentiality. They have potential, and they can regrow that arm. Other things are not as differentiated. There are some creatures that don't differentiate that much. Starfish, for instance. Starfish, you can take and chop a starfish in half, and it'll grow 2 starfish. Cut it up into 5 pieces, you'll grow 5 starfish. There's a limit, but that's because they don't have those many switches that they've turned on and off. Does that make sense? They're simpler, if you want to put it in a different way. And then you have a more simple organism called the planaria. Have you ever heard of a planaria? It looks something like that. It has little eye spots. Actually, kind of cute. But you can take those and you can and throw them in a blender, and you'll have a couple hundred planaria when they're done. They have each one of these has more potential, less differentiation, has some stem cells. This has more potential, even still, less differentiation, and this has the most potential, the least differentiated of these multicellular organisms as compared to humans. So the more complicated you are, the more specialized your tissues, the harder it is to go backwards, to go in the opposite direction, undifferentiate them, and get that potential. So my suggestion, you can talk to Miss Dalverin, if you ever talk to Miss Dalverin, she did this with her triplets. She had triplets, I don't know if you know that. What she did is she got her umbilical cord stem cells, and she had them saved. She had them frozen for her children in case that might help them in the future. So if you have babies, you can ask the doctors to save your umbilical cord stem cells, and they'll put them in liquid nitrogen. You could save them until your kids get old, in case they might be able to use some of those for some of these techniques that I've just been talking to you about. Does that make sense to all of you?
Mm-hmm.
All right. And then that would get rid of the idea of, because a lot of people have a problem with stem cells because some of the stem cells might have come from abortion clinics, etc. That would solve your problem, wouldn't it? If you saved your own umbilical cord stem cells. All right. So now let's talk a little bit about what we've been talking about all this time, which is proteins. How do we go from a gene to a protein? How much time do we have left?
It's
8:32. So we got some time. So we're talking about going from a gene. Where is this gene? Where is this? This thing is called, by the way, let's talk about what this thing is called. This is called, anybody know what you would call this thing? DNA molecule. Good. We say it's double-stranded, but a chemist would argue that these are actually 2 molecules, hydrogen bonded together. I'm glad you can see that, because a chemist would argue that these are 2, and there's a really good point to that. They are, because these are just hydrogen bonded to one another, and there's 2. There's a 3' end and a 5' end. And then they're just hydrogen bonded to one another. 3' and a 5'. And this is 5' and this is 3'. So they're complementary, they're hydrogen bonded, they're double helix, they form, but a DNA, we call it a DNA molecule just to make it simple and easy. That's why they call it a DNA molecule. So this is very long. If you looked at when DNA in your cells are all wrapped up, let me redraw this. If I drew your cell, if I looked through a microscope at any one of your cells, and we had a really good microscope and we knew what we were doing, you would see a lot of these structures. You'd probably see a few dots that would get, then you'd see this. If you zoomed in to your nucleus, that's where you'd find your DNA. You'd find your DNA, and I'm going to go ahead and draw the DNA as blue. You would see your DNA would look like this most of the time. it would look like a bunch of spaghetti. We call that chromatin. Chromatin. That's when the DNA is all unwrapped. It's all unwrapped, it's being used. It's when DNA is being functional. It's doing its job when it's unwrapped. So when it's most of the cell's life, the chromatin is all unwrapped. That ball of spaghetti is actually 23 pairs of DNA. One set of 23 from mom and 23 from dad. So for a total of what? 46. 46 chromosomes and 46 copies in humans. This is in humans, obviously. I hope you know it's in humans. In humans. Corn has hundreds. Corn has hundreds of these copies. There are some organisms that only have 3. So the number of chromosomes depends on the creature, on the species. So 46 copies. When not all of the DNA is unwrapped. Some of it's unwrapped, then some of it's wrapped up around histones. I'm going to go ahead and draw the histones in red. And then some more unwrapped, and there's some more around histones. What do you think is going on here? Why is some of it unwrapped and some of it not unwrapped? Yeah.
Some of it is being used like at the moment.
That's right. Excellent. So some of this DNA is being used. So this is used. That's excellent. That's really that shows you've been paying attention. That's really a good point. This is being used, right? All the stuff that's unwrapped is being used. So what about the stuff that's wrapped up? What about the stuff that's wrapped up?
It's not being used.
It's not being used. It's being stored. So we call it off, right? Not used. And so these are genes that are off. So these are the genes. This is how one way we can turn genes on and off is to keep them wrapped up around histones. We keep them put away. We're not using them, we're storing them. The ones that are being used are unwrapped. And you unwrap different sections of the DNA depending on which cells you're talking about in your body. But the DNA molecule, the sequence from one end to the other is the same. It's just your eyes, you wrap up certain ones, and in your foot, you wrap up another section of it. Does that make sense? That's just one way. There's several ways we turn genes on and off, but that's one big way. Are we good with that? Mm-hmm. All right, so there's 23 pairs, and again, you get 23 from mom and 23 from dad. And we'll talk more about that when we get into genetics next week. Next week, we're already in genetics, so. All right. This week, what you got to understand is in the nucleus, all in the nucleus, this is all happening inside the nucleus. DNA, and please know this by heart, is only in the nucleus. In eukaryotic organisms, I hope that's obvious. I hope to me, they seem obvious. But let me be very clear. In organisms with a nucleus, DNA is only in the nucleus. Did I make that statement clearly enough? Mm-hmm. Prokaryotes have DNA. This is a mistake that students make all the time. They think DNA is only in the nucleus. Prokaryotes don't have nucleus, so what do you think? They don't have DNA. Well, that's not true. They do have DNA. It's just not in the nucleus, okay? So don't get that confusion in your mind. Those organisms that have a nucleus, the DNA is in the nucleus. When it's being used, it's unwrapped. It's formed these things called chromatin. And again, the DNA is only found in the nucleus. Now, I should tell you about 2 other kinds of DNA. When we're ready to move and separate the cells, what do you do when you're ready to move stuff? You're working in a law office, and the law office has thousands of files and folders and cabinets all around. You have to organize them, and you're going to move offices. You're a lawyer, and you're going to move to another building. What do you got to do?
Pack them up.
You have to pack them up, right? Otherwise, you're going to lose them. You're going to lose them. Things are going to get confused, right? That's no good. So we have to take all this ball of spaghetti, and we have to make copies of them, because we're going to split them into two, right? So we have to make copies of them. And then we have to hold them together, make sure we keep them together, make sure we keep them straight so we can move them. So we need to pack them up. Well, when we pack them up, when we make copies and pack them up, this is what they look like. They look like this. They're packed up. They're made copies. These are copies of one. These are copies. They're stuck together. Keep the copies stuck together. They're like paper clips. Keep the copies together. And they're all packed up. They're no longer unraveled. They're 100% wrapped up. So they're wrapped up around histones in this tight little, sort of like braiding your hair, right? They're tight braids around, and they actually get real tiny, so tiny that you get 46 of these times 2, because there's 2 of them. They're copied, so there's 46 of these. We call these chromosomes. It's the same DNA. The DNA didn't change. They're just packed up. When you hear the word chromosome, you're just talking about the fact that you're wrapping it up. And you only see these wrapped up chromosomes after the copies. So you have 2 copies. So technically, you don't have 46 anymore. You have 92. And I shouldn't have said, so you start off with 2 copies of each chromosome. See, this is technically, each one of these is a chromosome, but when they're together, we also call them a chromosome. So together they're a chromosome. Once they separate, we'll also call them a chromosome. So don't get confused about that. You'll see that when we talk about mitosis and meiosis. But these 2 copies here, these 2 copies are called sister chromatids. We'll talk more about these later, but it's, I think, a good time to discuss these different terms. Yeah.
Isn't that one meiosis and the other one mitosis has like 4? Isn't it something like that?
Yeah, it's something like that. It's mitosis happens. We'll talk about it. I guess it's really kind of cool. I like mitosis. You should have learned a little bit about it in middle school, probably, if you had a chance, if you had a teacher that liked it. Mitosis and meiosis are very important. Mitosis helps you repair yourself when you get cut. Mitosis allows you to repair your cells. They replace cells that you hurt or damaged. Meiosis makes when you make a baby, you're doing meiosis to make your eggs. You have 10,000 eggs. Those eggs all were formed from meiosis. You take half the DNA. You don't have 46 copies in your eggs. You only have 23 copies. Only half the DNA. That makes sense if you think about it. If you're going to get together with your husband, you're going to make a baby, you're going to need... That's right. Excellent. 23 of his and 23 of yours, and you make one whole person, right? All right. But that's only for humans, because different ones and different creatures. All right, so unwrapped, they looks like that, and wrapped up, they look like that, and that's how you turn them off. So now the question is, what's happening when you're turning them on? You're taking this sequence. I'm just going to write just a quick example. Let's say it's A, T, G, C, A, T. That's the sequence. It's a lot longer. It could be hundreds, maybe even 1,000 nucleotides long, because these are very long. Understand, they're only a single molecule, but it could be as long as 3 meters. So it's really long. And then, of course, you wrap them up into this tiny little, all of it's all in one nucleus. So you can imagine how tiny these things are. So the first thing you do is what? What do you do? Oh, I should tell you, when you're talking about it, not all of the DNA is a gene. Only a certain section, only this part right here is going to be a gene. If this is the gene right here, if this is the sequence that's going to be turned, and by the way, when we're expressing a gene, when we're turning a gene on, when we call it a gene, that means this turns into an RNA. We don't call it, they're not genes unless they make RNA. Is that clear? Mm-hmm. All right, so the rest of the DNA is used to regulate. So you have this area before the gene. This area before the gene is called the promoter region. What do you think the promoter does?
We learned about it yesterday.
Yeah, you probably saw it yesterday a little bit. What do you think this little section of pink does here, this promoter area?
It helps run.
It helps. It promotes. It increases. Without the promoter, it's not going to happen. So I like to think of the promoter area as kind of the landing strip. To make anything happen, what is it that you need to have almost anything happen in a cell? Protein, a specific kind of protein called what? Well, they could be ribosomes, which are a mixture of proteins and RNA. But specifically, it's an idea with the enzyme. That's right. There's this enzyme complex. It's a complex. There's more than one thing. It's called RNA, because this is what it's going to make. It's going to make RNA. Polymer, because it's going to make a polymer. A polymer of what? RNA. ACE. All right. ACE means it's an enzyme. So you got this polymerase. It's called RNA polymerase. It doesn't really look like this, but it looks something like it's just this blob of proteins, different polypeptides. They need to come together and need to land here on the promoter region. So I just think of this as a landing strip for the enzyme. So the RNA polymerase comes in, lands on the promoter region, and then starts to read the DNA. The DNA has to open, so you get this thing that looks like this. There's two strands, so they have to open. And then there's a coding side, the side that's going to give you what that's going to be read. And then there's a non-coding side, the side that's not going to be read. So this is going to be read, and this is where you're going to see A, T, G, C, A, T. Now, if you looked at the opposite side, this would not be A, T, G, C, A, T, right? What would this side be? T, A, C, G, T, A, right? So this is the coding side. So that when the RNA polymerase lands and starts reading it, it's going to start making RNA. So this is going to start making RNA. So the first thing that happens is you get these things called transcription factors. That's another way we can control what turns on and what turns off. These little proteins in different RNA sections can land, and they're called factors. They're called transcription factors. This whole thing, taking DNA to RNA, is called transcription. It's called transcription. And these transcription factors, what they do is they help turn on those genes. Because you don't want these genes just turned on all the time. That would be a waste and also would be confusing for your body. You can't have every chemical reaction happening all the time. The whole point of life is organization. The whole point is you want to feel love when you want to feel love. You want to be angry when you're angry. You don't want to feel angry and love, hate, fear, bravery, right? You need to have one thing at a time. If you're feeling everything at once, you're just convulsing on the ground. You're not any use to anyone. Does that make sense? So you can't just go and stop. You can't just run and sit. That can't happen. So you have to have one thing happening at a time. So you need to control which ones of these genes get turned on and off. By the way, that's a really bad example because all those things were not directly controlled by a gene. But I'm just saying when you're trying to do a thing, you only want to do one thing at a time. You don't want to do everything at once. Does that make sense? So I use those examples. All right. And then at the end, you have what's called the termination sequence, right? So you have promoter, you have the gene, and then you have term. So the termination sequence, that turns it off. So So here's where the RNA polymerase lands, and this is where it gets kicked off. Lands, kicks off. Lands, kicks off. This middle part is what's being read. Then you read it, and you turn it into an RNA. Did I say mRNA?
No, you said
No,
RNA.
I said RNA. There's a reason I said that, so pay attention. So when I make this, it's going to be RNA. How would you transcribe this into RNA? It would be, would I make it
Opposite of
opposite A, which would be what?
T.
No. No T in RNA. No. Can't get matched with T. What does it get matched with? No. C only gets, with U. In RNA, you only form U. T takes the place of, or U takes the place of T in RNA. Right? I know it's a long time ago that we talked about it, but it's important that you know that. So you got U, then T gets read to what? Opposite of T is what? A.
Just U takes T's place.
U just takes the T's place. It doesn't take the A's place. So there is A in RNA, but there's no U. There's no T, right? So UA, then what? The G gets turned to C, and the C gets turned to
G.
And the A gets turned to
U.
And then the T gets an A. All right, and it goes on. It's a long piece. This is not mRNA yet. This is RNA. Then there's further control. Like there are other changes that have to happen to this RNA, and some of the things that have to happen is the introns have to be taken out. So there's sections of this that you're going to cut out. They're called introns, which never made sense to me because I always thought introns you should leave them in.
In because it says in.
In, right? But it's opposite what I think, but that's how I remember it. It's opposite what you think, Mendoza. That's how I remember it. Exons everything else is called an exon, and those exons, they stay. They get spliced together. There's a whole process we're not going to get into. There's a whole process. It takes the two sections that are not introns and it puts them together and makes one whole piece. And that piece has the U, the A, the C, the G, the U, and the A, and all the rest without the introns, minus the introns. It's still not quite RNA because the last thing we have to do is we have to add a poly A tail. We have to add a bunch of A's at the end. Once you add the poly A tail, you've taken out the introns, you add the poly A tail. Now this is an mRNA.
So mRNA is when it has the poly A tails?
It has a poly A tail and the introns have been taken out. Now I got to tell you, not all genes go to mRNA. Some genes get turned into RNA. Some genes, so let's take a quick look at what genes do. So yes, a lot of genes do go to mRNA. You can go to mRNA. We just went through the process. By the way, that's it. That's the end of transcription. That's all there is to it. Not that big a deal. Translation's a little more to it, and that's just taking a ribosome. We'll do translation tomorrow, and it's kind of fun. Most of the kids like doing it. It's like a puzzle. You have to read a chart, and just as easy as it is to go A to U and G to C, it's going to be very easy to read mRNA as well. So genes can go to RNA, mRNA, or they could just go to RNA alone. Things like tRNA, they can form ribosomes, and other structures that don't actually go to mRNA and make proteins. Remember, from mRNA you go to proteins. That's just a major path of genes. Not all genes go to mRNA and then to proteins.
So
All right.
what happens if it's not in that specific order?
Say that again.
What happens if it's not in the specific order of U, A, G, C, G? What would happen? Would it form something different?
Yes, absolutely. If you mess this up, you have a mutation. If it's here in the DNA, if you messed up the sequence here in the DNA, you have a mutation.
So, on RNA, the introns are still in there?
In regular RNA, when it's first transcribed, you still have the introns. Then something called a spliceosome comes in, cuts them out, and splices them together. Then adds a poly A tail. Now you got an mRNA. Now you're ready to make a protein. Hey, what's up?
Are they here to me Finley and
Taylor
Taylor Hudson?
Hudson and who?
Tyler.
I know. I'm sorry.
Yeah.
She said Taylor. I didn't. What is the other one?
To me Finley?
Finley. What do you need?
They just have to fill out a one-question survey for stats.
You have to fill out a one-question survey for stats. Are you here? Go to her.
It's time for us to go to the
All right, my friends, have a good one. You, Big Mouth, come here. What did you find during your research? I'm interested.
A lot.
Did you find anything that makes you think at all?
Some
Is
of it.
Some of it, yeah. Yeah, I found it. I found when I researched anti-vax positions, I also found it very interesting. There's some points that you need to consider when you're making those decisions. You know what I'm saying? But I want you to make I just play with you in class, but I just want you to think about it when you're making decisions. Don't listen to people. People are fools. When you're talking, you got to talk about facts and then make decisions for yourself. All right, have a good one. What happened to Emily? Where's Emily? What's going on?
All of them did. She was here today.
Well, there's a whole drama going on. I don't know what's going on, but I have to find out. I'm absent a few days, and everything gets turned upside down.
That's what always happens.
That's why I hate being absent. I'm doing all right. How you doing? Yeah, what's up?
So I figured I found out.
Don't be talking to me without a mask. Get away from me.
Bro.
Be 6 feet away from me.
Video on YouTube. Yeah, he gonna post it.
I'll try to post it today, yes.
You okay?
It's all right. It's hard, but you know how it is. I mean, I don't know if you know, but it's hard.
What? Why left my grandpa?
I hope you don't know. I hope you don't know for the many, many years to come.
I'm sorry.
It's all right, but listen, you're great. Make up everything you can. Just get it done. All right. If you need help, I'll be here during
I gotta
lunch.
be. I just need to.
Which lunch though?
The second lunch,
It's just
also
the
after
debate
school.
Stuff. That's the only thing I need to do. I did everything.
Debate stuff. We'll get this stuff done. Turn it in.
Did
I'll grade it. I don't care anymore. I'll grade it.
Did you see what I sent you?
No.
I had sent you a message because about my lab report. I turned it in with the first revision.
Why don't you see me during second lunch or after school? Quick though, because I need to go and get some stuff ready for the funeral.
I found out some more facts about the debate.
Oh, excellent. Well, I hope you're ready, baby.
She works for a hospital.
Excellent, but don't be late to class. See me after school, okay, baby? However, there is something we should really, I want to focus a little bit on, even though we're not there yet. I want to start you off with this idea of what we're talking about when we're talking about numbers of copies of DNA. So like I said, you have 23 chromosomes, and the chromosomes, and I'm going to go ahead and look at them as they're packed up. So as they're packed up, they're generally packed up after they've been copied. So these two right here, if this in mitosis, and we'll look at what the difference is between mitosis and meiosis in a little bit. But in mitosis, these two here are exact copies. They're the same. So whatever gene is here, so think about what that means. Whatever gene is here on this chromosome is here on this chromosome. If this is, and I know some of you did Punnett squares in middle school, even though you may not have done mitosis, right? So this is what the little a means. Little a would be this, so then the little a would be over here as well. When you learned little a, big A in Punnett squares, what they were trying to tell you is a mathematical method of analyzing how these chromosomes separate in a cell. So there's another, this particular copy might have come from mom. The mom symbol looks like this. Dad's copies, and I'll just go ahead and use the traditional little blue. Dad has the same chromosome, might be bent in a different way or whatever, but it doesn't matter. Dad has that same chromosome, okay? He also has an A, but you don't know if he has a small a. He might have a big A. Again, there's two copies here because this is after the DNA has been copied in this example. Is that clear? All right. So you only have, once the cell separates, you're doubling the DNA because you're making copies of the DNA because you're going to split the cell. Does that make sense? After the cell's been split, you're only going to have one copy of each again. But you will have one copy of each. So you have one big A and one little a. Does that make sense? Mm-hmm. All right. I hope you're not just sitting there quietly and not asking questions, or if you don't understand something or you're confused about something, now's the time to ask. So this person's genotype would be heterozygous and they would be big A, little a. That's what that means. That's what big A, little a means. And when you're looking at copies, think about it. Now I'm not gonna worry about mitosis right before. This is just before mitosis. This you copy it so that when you split the cell after mitosis, mitosis is a division of the cell, is a division of the nucleus, is a division of the nucleus. So let's look at after mitosis, you have two nuclei. You have one copy of the little a and one copy of the big A. One came from mom and one came from dad. Does that make sense to everybody? Yes or no? Mm-hmm. Each cell has a copy. So every one of your cells has exactly the same DNA for all of them. So that's two copies of this chromosome, the two versions. We call these two versions, these two chromosomes, homologues. The reasons, homo means the same, but they're not quite the same. So we call them homologues, right? They're homologous, they're similar. They have the same genes, but they might have different alleles. We call these things here, these different versions, alleles.
So when it's a big A and a little A, they're called
Alleles. The gene itself is the same. For instance, this gene might be for hair color. Let's look at hair color, or eye color. Eye and hair color and skin color is a bad example because color in humans is controlled by 6 different genes, in this case, of skin color. So you have like 48 or 64 different skin tones. So it's not black or white or brown. There's 64 different shades of skin color for humans. Eye color would not work either because there's multiple genes controlling it. However, blue eye color is recessive. So little a is, you would get blue eyes. So blue eyes are actually recessive. So you could be a carrier, you could be heterozygous for A and have brown eyes. So this actually controls whether you get blue eyes or brown eyes. Another example might be sickle cell anemia. Sickle cell anemia is recessive, so you'd have to have 2 little a's if you're going to have sickle cell disease, right? Both these cells have the trait, but in order to have the disease, you'd have to have both little a's. Does that make sense? Mm-hmm. So both of these control the same gene product, just they're 2 different versions of the protein. This protein would get you sickle cell if you had 2 copies of it, but because you have this version of the same protein, you have not sickle cell You have normal hemoglobin.
Mr. Mendoza,
Yes.
when I seen homo, I thought like homeostasis, if that's a thing, because you saw on the.
Homeostasis, yeah, that's right. Homeo just means the same. So you're going to see homeo a lot. And I think you'll see them in even the stores. I think they have homeostats they sell at the store and that kind of stuff. Homeo just means the same. And that's all.
Do that lesson tie in with this, like learning about mitosis and like homo?
It kind of does, because homologues means the same. They're the same. They have the same genes. Homologues have the same genes. They're not identical though. Sister chromatids in mitosis. I'm saying mitosis on purpose. Meiosis, it's a little different. But in mitosis, the sister chromatids are exactly the same. So this is exactly the same. They're identical. That's why we call them sister chromatids. I don't know why they call them sister chromatids. Why not brothers? Why not twins? I don't know. But they're sister chromatids. They're identical, okay? Homologues are not identical. They just have the same genes. They have the same, they code for the same proteins on each of these. Does that make sense? Homologues have the same genes, but they don't necessarily have the same alleles. So these are, homologues would be, in this case, for instance, this would be this, what the heck is it? Let's call this chromosome number 1. So chromosome number 1 in all humans, all right, has the same genes. We all have the same genes on chromosome number 1. It's just you might have a different set of alleles. You might have little a, I might have big A, which makes us slightly different. But we all have the same genes. That's why we're the same species on the same chromosome. And understand they have many genes on this. This is another important fact. They have many genes on this chromosome. I'm just looking at little a, but actually you'll have a B in here, and you'll have a Z, and you'll have an H, okay? And they actually have fancy numbers, but just for the sake of your Punnett square analogy, these genes, there's many genes on any one chromosome, all right? But they are always the same, and they're in the same place. So if there's an A here on a chromosome, there's going to be an A here on this chromosome. They're on the same place. They're the same genes. They might just be different versions. They might have these are mutations, by the way. This is what we call mutations. Little a is a mutant, and big A is a mutant. They're changes in the DNA. So that's just chromosome number 1. You have 23 of them. So you have another 1. Let me do it in white. You have another 1, 2, 3, 4, 5, 6. Oh, am I doing 2 copies? Yeah, that's right. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. I'm doing it wrong. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. I'm only at 11, so I have to make this nucleus bigger. It's going to be a weird looking nucleus. That's just the way it's going to be. So I'm at 11. (11), 12, 13, 14, 15, 16, 17. These are not connected. 18, 19, 20. Yes. 21, 22. Now 23 is different. 23, that 23 is the exception. There's 22 chromosomes are fine, but the 23rd, one is big and the other one is small. One is big and the other one's small. This is the X. There's 23 pairs. You got one from mom and one from dad. What you got from mom is always going to be the same. You can only get one type of this 23rd chromosome from mom, and that is this letter that you all are familiar with, X. Girls can only give an X because that's all girls have. Girls have 2 X's. Boys have a Y. So boys have a Y. So I have an XY. Males in the human species have an XY. All the rest of our chromosomes we all share. All of us have the same, pretty much the same genes on all the rest of the chromosomes. But ladies don't have the Y. Men do have all the genes women have, but we have extra genes. The Y that are on the Y, they're different. So Y is a different set of genes.
So a girl gives the X and a boy gives the Y. Well,
A
the
boy
male-
can't give an X or Y. You got an X from your dad, because we have both. We have an X and a Y. We can give you an X or a Y. You can only give an X to your child.
So a male can give an X or a Y.
That's why males define sex. We define whether you got a boy or girl, because our sperm depends which sperm. Does the sperm have an X? Because you remember, when you're making your sperm or your eggs, what you're doing is you're going to take this cell. This is in meiosis now. This is kind of a foreshadowing of what's coming. We're not covering this yet, but in meiosis, you would split these up into 2. In a guy, we would put him in sperm. All right. In a lady, you'd put him in eggs. But you don't have to. See how you have 2 copies of each chromosome? From 1 to 23, the last set being the set. All these are autosomal chromosomes. This is the 23rd chromosome is your sex chromosome. So we, as males, we can put 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. I'm going to have to redraw again. 21, 22, and then we have this last spot is going to be a little different. We're going to leave a little blank here. And this would make your sperm, right? So we have, notice here we have 2 copies of each gene, one from mom and one from dad. In the sperm, we only put 1 copy of each gene. And for the other chromosomes, it doesn't matter because they're all the same genes. And it does matter because you have either have 2 different alleles, if that makes sense, right? You can have a big A or a little a, or big B, a little b. So if you get this one, you got a big A in the sperm. If you give this one, you got a little a in the sperm. Does that make sense? Yes? Mm-hmm. Okay. Same with the ladies. The difference is when a lady goes through oogenesis and she makes the eggs, she can only give an X. So she's always giving an X chromosome. A guy is going to make one of his sperm is going to have an for every 1 sperm that has an X, let's make it yellow. There's another sperm that's identical that has, instead of an X, it has a Y.
So every X there's a Y.
Yeah, a boy. Now, yeah, that would if this is the sperm that met your mother's egg, you're a boy. If this is the sperm that met your mother's egg, you're a girl.
You said that before.
Yeah, so that's it. It's a 50-50 chance. So just by odds. Now, it turns out that it's not as easy. There's something to do with the woman's vaginal fluids. It seems that I think the X can swim better in thinner fluids, or the Y can swim better in thinner fluids, or something like that. I'm not sure exactly what the details are there, but so the female does have some input on whether they get girls or boys, and that consistency of the vaginal fluids can change depending on diet or circumstances. So it'd be interesting to see if there's a correlation between environmental factors, like maybe in times of starvation you get more boys because the guys can go out and take care of themselves faster. I don't know. I have no idea how it would work. But the point is that it would be interesting to see if there's a correlation. But just statistically speaking, the two sperm, the X and the Y, men make 50% of their sperm are X and 50% Y. So 50,
Even number.
just even number, because they have one. You're taking half of these randomly and putting them in two sperm. Does that make sense?
Yeah.
So things are a little more complicated than I'm saying, and we'll go over it when we go to mitosis and meiosis later. But just so we're clear, when we're talking about this DNA, we're talking about these chromosomes that are in the nucleus. That's what we're talking about. We're talking about it's all connected. I think too often we separate things, like we're going to do Punnett squares today. Yay, we understand Punnett squares. And then we go and do cells. Yay, we did cells. And then we go and we did protein synthesis. Yay, we did protein synthesis. But all too often we separate them, and we don't think that they're actually all the same thing. They're all connected. All right. So let's talk about protein synthesis, about transcription. Let's go over what we went over. Let's go over what we went over yesterday, and let's see. So we said we have this DNA, and we're just going to look at a gene, and we have this area that's a promoter region. We're going to remember that this promoter region is what's going to allow you to regulate that gene. Now, I'm only looking at the gene. You have to remember this is very long. It can be very long, many genes. Some of it's wrapped around a histone. Remember that. So you can turn some genes on and some genes off. Don't forget that, because that's an important regulatory step. Some of the DNA is wrapped around, it's not going to let loose. But then you have the gene. Now you have this gene, it's in this particular cell. This is what we want to express. We want to make it into a protein. And then you have at the end of it, you have a termination sequence that says that where it stops. It stops it. So this is the coding region. So how are we going to do it? What are the steps? If I gave you this on a test and I said, make me the word here, what word are we going to use? We're going to use the word transcribe. Where is this happening, by the way? Where's transcription happening? Where's the only place that DNA is found?
Oh, in nucleus.
In the nucleus. In eukaryotes, this is happening in the nucleus. What about prokaryotes? Where's it happening? In the cytoplasm. There's no nucleus, right? So it's just happening in the cytoplasm. And the other thing about prokaryotes is their DNA is circular, so there's no 3' end and 5' end. They don't. It is double-stranded, so they have to open it up like we do. But other than that, that's it. Yeah.
What is the purple and the red boxes say?
Promoter on the purple and termination on the red. So what's the first step? What do you need in order to do just about anything in a cell? What is it you need?
Protein?
A protein. What do we call that protein?
Enzyme?
Enzyme. Excellent. You need an enzyme. Who said that? Excellent. So you need an enzyme. What enzyme is going to make RNA? It probably, remember how we name enzymes. It probably has something to do with what we're making. So we're making RNA, so there's probably RNAs in it, and it is. And we're making a polymer of ribonucleotides. So what are we going to call it? Poly something, right? Polymer. And how do we end our enzyme names?
ASE.
That's right, ASE. So it's RNA polymerase. That's the enzyme, RNA polymerase. If we were making copies of DNA, what do you think we'd call it? If we're making a polymer of DNA, what would we call it? Polymerase, yeah, but would it be RNA polymerase? What would it be called? DNA polymerase. See how easy that is? So it's really easy. I know we think that science is supposed to be difficult, but we actually try to make it very easy. So RNA polymerase is going to come in, and how does Excel tell the RNA polymerase, hey, it's time to make copies of this gene? Well, we have these things called transcription factors. It's more complicated than this, guys, but I'm just going to go ahead and and just make it as simple as this. They're these little, those of you that play PlayStation would recognize these, right? So they're these little factors. Now these factors could be all kinds of things, proteins, maybe they're ions or whatever. But these things are gonna bind to the promoter. And when they bind to the promoter, when that happens, it's gonna attract the RNA polymerase. It's gonna tell the RNA polymerase, hey, yo, land here. So this is, I like to think of this as like a runway at the airport. And I like to think of RNA polymerase as the plane. Maybe that's not a good analogy, but that's how I think of it. You can think of it any way you want. Maybe a race. Maybe this is a raceway entrance, and this is a race car highway entrance, and this is the car, and this is the I don't know, whatever. But the promoter only is going to be extra active when these transcription factors are bound to it. So when these transcription factors are bound to it, then the promoter region is going to be extra sticky, if you will. So we got these things here. They're all bound to it. Now this thing's ready to take on the RNA polymerase. So here comes, and we're going to go ahead and draw RNA polymerase in a second. Let's do a different color. I'm going to draw RNA polymerase like this. It's really not like that, but let's go ahead and draw it. So it's going to land, and it's going to be sitting around the promoter region, ready to do its job. And as soon as the last transcription factor binds, it's going to start. It's going to start opening this DNA up, and it's going to start reading it. And it's going to start making. You have to understand. Somebody in my AP class actually asked me this question. I thought it was a good question. How does the RNA? How does the RNA polymerase find the RNA? The RNA's. This is a solution. We're talking about a solution here, right? So the solute here in the solute is all this RNA. There's A's and T's and G's and C's, and there's a lot of ATGCs. And U's, T's, A's, U's, C's, G's, they're all over the place, all right? So you got to understand that when we're talking about nucleotides, they're part of the solution. They're the solutes in the solution. And they're all over the place, all around it. They're not going to limit the number of ATGCs. They're not a problem. They're all over the place. As soon as this enzyme gets a chance to use them and put them where they have to go, it's going to do it. Good Lord, this girl. So, this thing's going to start reading. Now, what if this thing reads A, T, G, this is DNA here, T, T, A, T, G, C, A. What is this going to, it's obviously going to open up the DNA. It's going to start reading that. This is what it's reading in DNA there. What's it going to make? RNA, correct. So what's it going to, if it's reading that, what's it making? Show me the RNA it makes.
So like the letters?
Yeah, tell me the letters that it makes.
It'll be U.
U?
U, A, C, G.
Not C.
Oh, G.
G?
No, A. Because he wrote it differently from last time. U, A, C, G, U.
So if you can do that, if you can match them up and you can do that, you're already ahead of the game. That's transcription. Boom.
Transcription is just turning from RNA into
From DNA into
I mean,
RNA.
DNA to RNA.
All right, that's it. That's transcription. That's it. You just did it. That's all there is to it. That big word, and it happens in the nucleus. Happens in the nucleus, and there's all the reagents are necessary there. Then when it gets to termination, it stops. It falls off, and it either lands or not, depending if the transcription factors are there. All right, so you made this RNA. Now this is just RNA. It's not mRNA yet. Now it has a choice. What are at least three choices this RNA, depending on the conditions of the cell, what the gene is, all kinds of signaling stuff. What are three things that could happen at this point now that you have RNA?
It could turn into rRNA and
That's right. It can turn into rRNA.
mRNA.
It could be mRNA and go this way,
Or DNA go back to
or it could be tRNA, right?
Could it go back to DNA or no? Once it's switched.
That's a really good question. In humans and most animals, no. Once you do, you can't go from RNA to DNA. But viruses like HIV have an enzyme, a protein that can take RNA and make DNA.
So if you got HIV,
You have little viruses that take their DNA, their RNA, their inheritable information is in RNA. And then they have this little protein that they make that reads that RNA and makes DNA. And then that DNA makes a lot more RNA, and then that RNA makes the proteins, makes the virus, and away it goes. It's really weird. Life is not straightforward. What we're learning here is how your body normally does things, but there's a lot of exceptions. Things aren't there's very few things that are as simple as we tell you. I know sometimes you think I'm making things hard, but I'm actually trying to make something that's very complicated. Life is complicated. I'm trying to make it very as simple as I can. And as you get older and take more classes, you're going to be like, oh, that's what Mendoza meant. Or that's, hopefully you remember me, but whether you remember me or not, that's what I understand now. Or, oh, I can't believe it's that way. It's all kind of, like, I can't tell you how shocked I was to find out that ducks nest some ducks nest in trees. Like, I couldn't believe it. I was like, wait, a duck with webbed feet is going to make a nest in a tree? I know it's stupid, but it just shocked me.
You wanna know that?
No, most ducks make nests on the ground.
It's divers and dabbers.
Okay, you know more than me about ducks. Okay, but my point is that it shocked me. It shocked me.
Bird watch.
Yeah, I didn't know that. Hey, bird watching is cool. I wish I'm a photographer by hobby, although I haven't gone out. I gave my camera to my nephew. I haven't been out for so long. But I used to go take all kinds of photos. Oh, birds are cool, man.
No, we have an eagle nest by our house.
Oh, that's neat. I'd be scared though, because eagles are big.
They're huge.
They can pick you up. They can pick you up and take you and eat you.
Absolutely.
No, they can take. I've seen. Scientists have taken dummies that were 150 pounds, and put them near the eagle's nest, dressed them up like a human, and the eagle picked up the dummy. Granted, the dummy wasn't fighting back, right? But picked up the dummy and took it to the chopping block and cut its head off.
Wow.
I'm like, nope.
I weigh 115 pounds, so they definitely would be.
Yeah, I don't know if they could. I don't know if they would, but just don't. They're wild beasts, okay? So you have to be careful.
They don't really come down. Like when I walk my dog, you can kind of.
Do you have a small dog?
No, I have a kangaroo, but my dog is huge.
Okay, I say if you have small dogs, small dogs for sure. Eagles are gonna be.
Oh, duh.
All right. So you can make RNA. Now the interesting thing about this is that from this RNA, from this initial RNA that you transcribed or your body cell transcribed, if it goes to tRNA, tRNAs look something like this, and we're going to talk about them in just a minute. To me, they look like T's. I will talk about what. Huh? And tRNAs, I'll tell you what they are in a minute, but on one end, they're going to carry an amino acid, and on the other end, they're going to have a message. And the other end, they're going to have a message. In this case, AUG. Well, we'll talk about it in just a second. I just want to finish transcription. And then when you're making rRNA, this rRNA, what it is part of a ribosome. So there's different parts. You have a smaller subunit and a larger subunit. And there's more than one part to the subunit. But together, these two subunits are going to come together and make this thing called a ribosome. All right, so these two, number one and number two, they're part of translation. They're going to help make the protein, but that's outside the cell. All these are going to go outside the cell. The mRNA, yeah, they're all going to leave the nucleus, not outside the cell. I'm sorry, outside the nucleus. So this is all in the nucleus. This is all going to leave the nucleus. Now imagine this is happening 46 times. It's kind of crazy. There's 46 copies of the chromosome, right? 46 chromosomes, 23 pairs. So there's 46 chromosomes. They're all unwrapped, so they're all... It's a whole process. I'm just making it as simple as I can. So it's very long, but it's quick. I'm going to show it to you. We have some animations. I hope the internet's working today. It wasn't yesterday. So we're going from we got the now we're going to make the mRNA. How do we make the mRNA? We got to take this message, and we have to cut out introns. So you're going to take that U, A, G, A, U, A, C, A, C, G, U. And we're going to take this, and we're going to cut out pieces. And let's say we cut this out here.
So it doesn't matter what we cut out.
Well, it matters. There's a reason for that. It's a very specific. It does matter. I'm just making it up. For this example, I'm making it up. It doesn't matter because it's just an example. But it does matter because depending on what you cut out, it's going to change the protein. It's going to change the message. You change the protein. Again, remember, one change in one amino acid in your hemoglobin makes the difference between sickle cell disease and just having the trait, or sickle cell disease, or normal hemoglobin, right? So it does matter. It's a big deal. But the bottom line is you're going to make this shorter. So you're going to take out these introns. You're going to put the exons together, and you're going to make U, A, G, U. So this is your message just going out. This is now your mRNA. Remembering I'm going to tell you to remember it's longer. I'm just using this example. So now you have mRNA. This is your mRNA.
mRNA is just when you just take out your enzyme.
You made some changes. You made some changes. And you added, don't forget, you added a poly A tail. So you take out the introns, add a poly A tail, now you have mRNA. If you go, now If this RNA is going to make a protein, it always makes a protein. If this RNA is going to make a tRNA, it always makes tRNA. And if this RNA makes an rRNA, it always makes RNA. The gene does the job it's supposed to do. The message in the DNA tells the cell which way to go. All right, and the introns are decided by factors in the cell as far as there are alternative ways to splice this. You can splice it one way when this is happening, splice it this way when that's happening. And that allows your body to adapt to different environments and allows us to be very complicated individuals. We are much more complex than the number of genes which suggests we would be. The reason that increased complexity has to do with the different ways we can transcribe and then translate the genes. Yeah.
I noticed that all three of those things got RNA in it. Why is that?
Because they're all RNA.
So what does
They're all made of ribonucleic acids. They're not made of deoxyribonucleic acids. They're all made of ribonucleic acids. Remember that? Remember that the ribose has an oxygen on the second carbon. So They're all made of RNA. That's a good question, though. They have 3 different functions, but they're all made of RNA. The mRNA is just taking the message out. But they've all been transcribed. They all went from DNA to RNA. Now, if this is going to make a protein, we're going to go out of the nucleus, and either in the cytoplasm, if it's a protein that's for the cell, or in the endoplasmic reticulum, in the rough endoplasmic reticulum, RER, if it's going to be leaving the cell. It's either going to go into the endomembrane system if it's leaving the cell, or it's going to stay in the cell. Again, I'm making this much simpler than it should be. Our mRNA is going to go to one of those 2 places. In both places, you're going to have a ribosome. You have the 2 subunits, the big and the small. And there's a lot of these all over the place. You're just waiting for an mRNA to come out. mRNA comes out, and here's you have a U, A, G, U, A, U, G, C, A, U. I'm just making it longer just so that we can have something to work with. C, A, U, G, A. Oh, I wonder if I still have a code. Let me pause. Here we have this code. I made it longer than I had inside just for the sake of room. We're going to use this universal code. You know what's another interesting fact? All life on Earth uses the same code to translate mRNA. Isn't that interesting? The way you make proteins is the same way a bacteria makes proteins. The same sequence. It's the same as the tree that's outside in the courtyard. The same as the fish that swims in the ocean. The squid, the tube worms at the bottom of the ocean. All use this code. Isn't that interesting? Common ancestry, right? Because if it didn't have common ancestry, wouldn't you expect it to be different somehow? It's slightly different, but basically the same code. All right. So now what's going to happen is that you're going to sit here and as soon as this could be longer. This could actually be, this could be longer. It probably is. But this code right here, this AUG, is very important. AUG is the start of the protein. What happens is that you have the big subunit, has 3 spots. This is the ribosomal subunit. And the smaller one, there's 3 spots, 3 what we call catalytic sites that define 3 units of 3 on the message. These are the words in the language. So these are a bunch of letters, but we can take these and divide them into 3s. A triplet code. Triplet. Write it down. 3s. We call this the codons. These are codons. This is a codon. The next codon, I'm going to alternate blue and yellow so you don't mix them up. There's another codon. The next one is here. The next one's here. The next one is here, etc. Okay? So I'm showing right here, I'm just showing 1, 2, 3, 4, 5 codons. But obviously these could be hundreds of nucleotides long, so it could be 50 codons, right? So it could be anything. So as the ribosome reads these, 3 at a time, the codons are triplet codes, so there's 3 at a time. There's a matching anticodon. So if these are codons, there's a matching anticodons. And where's the anticodon? The anticodon can be found on the tRNA. And the tRNA, remember, is carrying an amino acid, some amino acid. It's a horrible drawing of a tRNA, but anyways. So that if the codon is AUG, what do you think the anticodon is going to be? Remember, anti means opposite. Is it T? It's UAC. UAC, good. So it's UAC. So that's the anti. Now this is the anticodon. By the way, this process that I'm describing right now is called translation. All right, this is happening in the cytoplasm or in the rough ER, one of the two. You will always have this chart. No one's going to expect you to memorize this chart. Is that clear? But you're always going to have this chart. And I'm going to give you an activity to do this weekend. It's going to have you practice this. And we'll talk more about this on Monday. So AUG is always the start. This is what we call start. Let's find the start here. Where's AUG? How do you read this? The first is where? Where's the first? AUG. Where's the first? Yeah. Here, the A, right? So this is the first. Bless you. Yes, this is the first. These are the first letters. So if the first letter is A, that means it's somewhere in this row. A. It's in A row. That's the first letter. Then U is going to be where? AUG. Wake her up. Yeah. Go ahead. Right here, right? That's right. The column, the first column. Third row, first column. So somewhere in this box, AUG. Don't close your eyes then. AU is somewhere in this box. Where's the third position? G, AUG. Right here, right? So what is it? What's AUG? It's methionine, and methionine is also known as the start. It's the start of transcription. All right, translation. Sorry, translation. Start AUG. You will. The first AUG in the mRNA is when the protein starts to be made. So this is not just any amino acid. This tRNA is carrying what? We just looked at it. What? Methionine, that's right. This amino acid is carrying MET. Methionine. This tRNA only carries methionine. Is that clear? If I see one more person with their eyes closed, I'm going to have you stand up. So when we're talking about translation, every tRNA is different. So what's the next codon? So we got the first amino acid comes out. It is methionine, MET. It's going to be connected to another amino acid. What was the second one? UAA. Is that correct? UAA. Let's remember, we're reading this according to the codon. We're not going to read it according to the anticodon. So we're only reading it according to the codon. So what is it? Out of curiosity, what is this tRNA? What does that tRNA look like? Oh. Sorry, give me a second. I gotta erase this. Wish I could make the eraser bigger, but. So this tRNA is the second tRNA. It has to be in the cytoplasm as well. The second tRNA has to have a different anticodon. The codon is UAA. What do you think the anticodon is? UAA would be AUU. All right. So that is carrying UAA. How would you read this? Please tell me what is the amino acid that this tRNA is carrying? UAA. UAA. Ooh, it stopped. What does that mean? What does it mean to stop? UAA means stop. You see that? What does that mean? What do you think it means? Come on, quick, make a guess. It stops. It means it stops. That's it. So this is a bad example, because then this protein would only be what? One amino acid, only be methionine. That's it. There is no protein. So this is one way you can have a mutation that stops the protein from being made. So someone like me that has an O blood type that doesn't have the surface proteins, right? Maybe I have a mutation that starts with UAA. So it doesn't make any protein. Does that make sense? So where you have an A or B blood type, maybe your blood type, instead of UAA, maybe you have this. Maybe you're just one position. All it takes is one position. Instead of UAA, maybe you have UAU. UAU. Look what happens if I just change this UAA to UAU. What happens? UAU, I have methionine, still the first one, right? Then UAU is what? What's UAU? UAU is what? UAU is TYR, which is tyrosine. So this is tyrosine. So then this anticodon, not U, it wouldn't be AUU, right? Because that's not, it would be AUA, anticodon. Would have TYR, would carry tyrosine. So your second amino acid would be tyrosine. All right, have a good one. They would have this amino acid called tyrosine here. So it would be methionine and then tyrosine. Do you see that? So this thing would ratchet up. It would ratchet up. You'd have methionine. This is the first amino acid that they would have, MET. And then that would be connected by a peptide bond to an amino acid called tyrosine, TYR. And that would be connected to another amino acid. We don't know what that amino acid is yet, which would then be connected to another amino acid. We're making a chain of amino acids. So AUG is methionine, UAU is tyrosine. What is GUC? You tell me. Now, you're always going to have the chart. You don't have to memorize the chart. So you're always going to have the chart. Wait, what was it? What did I say? What is it? GUC. GUC is alanine. Is correct. GUC is valine. VAL. Are we good? So the next one's valine. Do you see how easy this is? It's really easy. And you would just keep reading it. There's AUG again. There's methionine. So you have methionine, tyrosine, valine, you have methionine again. You see AUG is there twice in the beginning, and then it's there again here. And then you have CAU. What is CAU? I don't know. I'm looking it up. Go to the chart. CAU is what? Histidine. So then you go ahead and add histidine. And then you have G, A something, etc. Until you get what? Until you get one of these three. What three is going to stop? What are the three that will stop it? UAA, UAG, and UGA. Those three will stop it. Are we good? That's all there is to translation. That's all. It's really that simple.
We're gonna be tested on that.
Yeah, transcription translation will be tested on. That's gene expression. Go back where? That's all there is to it. It's really easy. I think it's easy to read the chart. Each piece is easy, but if you don't put it all together, it can get very confusing. Am I doing transcription? Am I doing translation? Am I making RNA? Am I making a polypeptide? Am I reading DNA? And what is going on? Yeah.
How do you know what goes against what though?
What do you mean?
Like you see, but what goes against that?
GUC. What matches GUC? Well, all you got to do is read GUC. You don't have to find the match. All you got to do is find G, U, and then C, valine. That tRNA will carry valine. The tRNA that matches GUC will carry valine. And so you can come up with the list of amino acids given any gene. I'll give you a DNA molecule. You'll transcribe it to mRNA, and then you'll translate it into a polypeptide. You'll do that. That'll be your activity for the weekend. It's easy. It's fun. It's actually kind of a fun little puzzle. All right? Then I'll give you, for next week, on your last, I'll have them ready for you by Monday, I hope. You'll have, certainly by Tuesday, you'll have your weekend voluntary practice for your final. Everything on that practice will be on your final. If you need to go back, read the book, do whatever you got to do to get ready for the final. But I'll give you that study guide for you to, and if you do it, you'll get points. If you don't do it, you don't lose points. You just don't get the points. Yeah.
So this is all tRNA.
All right, so tRNA comes into the ribosome. This is mRNA. This is tRNA. This is rRNA. So there's 3 kinds of RNAs involved here. That's a good question. Let's make sure we understand that. Please understand that this is tRNA here. This is rRNA, ribosomal RNA. And this is mRNA. Is that clear?
Yep.
And then the thing that you make at the top here, this is amino acid sequence, a polypeptide. This is a polypeptide. So 3 types of RNA come together, tRNA, rRNA, and mRNA, to make one series of amino acids connected together called a polypeptide. Are we all good with that?
Yeah.
All right. And where is that happening? In the cytoplasm, the RER. Where does transcription happen? In the nucleus. It's just that simple. And by the way, this is going to, I can almost guarantee you it's going to be on your end of course exam. You're going to see it again. Pretty simple. If you see this, you'd be like, yes. If you see this, osmosis diffusion, you're like, yes. Is the cell swelling? Is it shrinking? Yeah.
All that stuff is outside the nucleus.
All this, that's right, good question. All this is happening outside the nucleus. All this is happening inside the nucleus. So transcription, taking DNA, making the RNA, has to happen in the nucleus because that's the only place you have DNA. Yeah. Besides the mitochondria.
So does it move around? Like it's in an open area?
Yeah, there's little nuclear pores that let it out.
It moves around it though?
Do they move around? This is happening
That.
what this?
Yeah, it moves around the nucleus.
This is happening all over the place. It leaves, this RNA leaves here, maybe another one leaves out that way. So that you can get multiple copies of mRNA making multiple polypeptides throughout the whole nucleus, or the whole cell. What?
Where does it go?
Where does what go?
It don't go. You said it leaves. Where does it go? Just
It leaves the nucleus and goes into the cytoplasm, into the space out here that's still inside the cell but outside the nucleus, or it goes into the endoplasmic reticulum. Either way, depending on whether the protein is leaving the cell, being excreted like mucus or hormones, or whether the protein's staying in the cell. Does that make sense? It's good, very good questions. And leads through the nuclear pores. I wanted to show you a movie that shows all this to you, but the internet's not working, so I'll have to post it. And I'll have to put in another ticket to see if it'll actually start working. There's a bunch of videos. It's a really cool video that shows you the amino acid being built. Another one that shows the protein, the RNA being built. Another one that shows the actual protein leaving the nucleus and going to the ribosome.
is it moving as fast as it goes in the body?
Oh yeah, it moves. It shows you real time. Yeah. Obviously, it's computer generated because you can't see this, brother. This computer's messed up. This one, I'm trying. What Wi-Fi are you